can start now. Yeah. Yeah.
Hi everyone. So I'm gonna share my screen now and start my presentation. Okay. I will talk about optomagnetic biosensing for nucleic acid-based diagnostics, and I will give a broad introduction of why is it important and how it contributes to the big picture of the diagnostics research. And on this slide, I picked tuberculosis as a reminder that it's not only new diseases that are popping up every year, but it's also forgotten diseases like tuberculosis that are still a significant health concern, especially taking into account that it's a respiratory disease, so it can be spread, can come back. So let's speak about diagnostics. At this time, the world is threatened by a COVID-19 infection. And here in Denmark, they built this, what they call TELT. It's like temporary tent where you can get diagnosed or can get tested for COVID-19. But it's something new because normally if you would like to go for diagnostics of HIV or hepatitis or I don't know, any other disease, it's just normal clinic without need for such temporary tents. But in other countries, such as for example in Africa or Central America, where there is lack of centralized labs and hospitals, such in-field diagnostics is quite common phenomena. So you can see these doctors sitting in the middle of the field and doing the tests. So that's what we call point-of-care diagnostics. And we would like, of course, that diseases like tuberculosis can be diagnosed also in field, so that point-of-care solutions can be developed for such diseases. So HIV is a classic example to introduce how point-of-care diagnostics work. In this slide, you can see that blood is collected from a finger by a low-skilled technician who just needs to mix this with solution number 1 and then pour the liquid from this little bottle into the membrane. And then he needs to pour liquid from bottle number 2 and from number 3. And then he, together with the patient, they look onto the membrane and guess what they see? If they see only 1 point, that means that the test works fine and the person is negative for HIV. If they see 2 points, it means that the test worked fine and the person is positive for HIV. If they don't see any point at all, it means that the test failed. So the test needs to have internal control so that we know that it's working. And another signal will come whether the result is positive or negative. So this is what they call it's not even quantitative, it's only qualitative test, but it's good. You can find this point-of-care diagnostics of HIV in all developed countries, because people would like to, of course, eliminate or vanish this disease. So why not to have similar tests for other diseases like tuberculosis or dengue fever? The thing is that HIV can be prevented by practicing common sense, whereas respiratory diseases, unless a certain action is taken by government. You cannot prevent spreading respiratory diseases because of the migration, traveling, holidays, and refugees. So it's rather uncontrollable. So we need to find a solution where the even latent form of disease can be detected. Latent means that the person is infected, but he doesn't have symptoms. So such person can spread disease even faster and to more severe extent than the person with symptoms, who obviously will stay home until the symptoms are cleared. On this slide, you can see a double-stranded helix of DNA. And I will remind that there's made of certain nucleobases and it carries genetic information. In a database, you can find the genomic sequences of any organisms, bacteria, viruses, and animals, humans. And thus, if we talk about bacteria and viruses, knowing the sequence of genome, we can design primers to amplify these sequences, even if there is only one molecule of such DNA or RNA floating in a blood sample or in a sputum from the mouth. And this is a classic, the most gold standard of molecular diagnostics is called polymerase chain reaction, in which you can see DNA sample is mixed with DNA polymerase, buffer, primers, nucleotides, and placed in a thermocycler where after 20 or 25 cycles of temperatures, 95 degrees, 55 degrees, and 72 degrees, you get exponential amplification of the number of the DNA or RNA copies. So it's an extremely powerful technique. And the question is why people still do research in molecular diagnostics if the problem is solved. Yes, you can detect few copies of nucleic acid material through the amplification. However, the equipment and the complexity of temperature cycling, it must be really precise. Otherwise, it will be really messed up if the temperatures are not as they are stated. And the changes between the temperatures should be also within seconds. So such diagnostics is implemented in central labs, in centralized hospitals, and in the well-developed countries. In fact, the COVID-19 test negative is performed using reverse transcriptase PCR because it's RNA. So RNA needs to be converted into DNA and then DNA is amplified. So now if you go to be tested, if you're going to be tested for COVID-19, you will know what does it mean. It means the negative result means that they could not find any copy of RNA molecule in your sputum. An advantage of molecular diagnostics is that it can be detected rather early. So coming back to the HIV example, you can see on day 0, there was a potential exposure to HIV. After 1 week or 7-10 days, RNA can be detected using polymerase chain reaction. After 16 days, P24 antigen can be detected. And after 28 days, the 4th gene antigen antibody test can detect HIV. And after 90 days, antibody test can detect HIV detection for sure. So the very first slide where you have seen this point of care diagnostics using membrane and 3 solutions and looking into the membrane, whether there's 2 dots or 1 dot, that can detect the disease after 90 days. If you would like to detect it earlier, then you have to perform appropriate tests. And the best is molecular diagnostics because the RNA copy can be found after 7 to 10 days. Now, speaking about the target, the same symptoms can be often caused by different pathogens. For example, two respiratory diseases, tuberculosis and dengue virus, and on the top of it, influenza and other, like pneumonia, other respiratory diseases. You need to be able to understand what is the cause of the problem. And on the top of it, dengue virus, for example, has 4 serotypes, and the outcome and treatment will be depending on which serotype is found. They are very homologous, so only nucleic acid test can say whether it's serotype 1, 2, 3, or 4. And for Mycobacterium tuberculosis, a mutation can cause antibiotic resistance, so that the disease that can be treated with antibiotics is no longer susceptible to the antibiotic. So even if the tuberculosis tests, which we will discuss just after this slide, if the tuberculosis test is suspicious or positive, before starting treatment with antibiotic, the doctor needs to make sure that there is no mutation and the disease is actually susceptible. Otherwise, only harm will be performed to the patient. Yeah, accordingly to the World Health Organization, tuberculosis prevalence in 2014 was 42% lower than in 1990. So thanks God, the problem is not that bad. However, an estimated 9.6 million people worldwide developed active tuberculosis in 2014. And in this case, a co-infection like HIV or diabetes significantly makes the prognosis significantly worse, prognosis of the disease. The estimated incidence in Europe represents 3% of the global TB burden, tuberculosis burden. However, we know that the life standard in Europe is good. So the main problem is in poor countries with lack of centralized hospitals and labs, just as I mentioned, where we need to build these tents and bring point of care solutions where we can diagnose disease. So personally, in Russia, we were exposed to this tuberculin skin test, where they inject tuberculin under the skin of the forearm, and then they measure the size of the, how it's called, little thing that grows on the skin. And depends on the condition. For healthy people, if the thing is bigger than 15 millimeter, the person should go for the further checkups. But if the person is known to have HIV or other risk factors, then even lower, smaller size of the thingy is a sign for him or her to go for further checkups. Yet the test is very unspecific. In my case, it was always more than 2 centimeters. So, and I never got tuberculosis. But what else they look at is, of course, is lungs, fluorography or X-ray. But usually, if the doctor can see changes in the lungs, it's already quite late. So we would like to develop solutions not for advanced developed tuberculosis, where every test is starting to be positive, but you would like to detect rather at early stage, and in particular, latent tuberculosis, as well as resistant tuberculosis. So when the person is sent for additional checkups, they usually make culture test from the sputum. So it's a standard microbiology test when they take a sample of sputum and smear it on agar plates, and they grow them without or with antibiotic. However, tuberculosis grows extremely slow, and it takes 6 to 8 weeks before a negative result is reported to the physician. On the picture, you can see on the left side, the strain is susceptible to antibiotic. On the right side, it's not. And this is how they know which antibiotic they need to give to the patient. It's very important. Giving the wrong antibiotic that is not working will only make harm. How else can we test tuberculosis, like to find tuberculosis and test it for the resistance? Use molecular diagnostics, because it is well known that the target that is binding antibiotic and leads to bacterial death can have a mutation, and the altered target DNA with a mutation will not longer bind antibiotic, so the bacteria will resist. And as we all know, DNA is a genetic code. So once mutation happened, all the all the secondary and further generations of the bacteria will carry this mutation. So that's how the bacteria exist. In tuberculosis, they found several genes that are responsible for the antibiotic resistance. And 2 of them were quite successfully implemented for diagnostics purposes, called CATG and RPOB. I will speak about them later. So we are still in the introduction, but now I will introduce you more of nucleic acid test and how it works. This is a real-time PCR machine. What it does, it has a thermocycler inside to perform cycles of the temperature necessary to amplify copies of DNA or RNA. And at the same time, it has a fluorescence reader that detects fluorescence. As more DNA is amplified as a stronger fluorescence signal. And you can set the threshold value, the CT value. And if the signal is above CT value, then it's positive. And if it's below, then it's undetectable. And usually you also run no template control. So it looks perfectly. You have a positive, negative control, and you get the answer from the machine, which is completely automated and user-friendly. However, the price of real-time PCR systems is ranging from somewhat $15,000 to $90,000, and you know, it's better quality, higher price, so to say. So this makes these devices good for centralized hospitals and well-developed countries, but completely unacceptable for those point-of-care solutions that I was talking about, where you can bring machine to a poor country and screen people for tuberculosis or other infectious disease. How else PCR can be read out? So this is a multi-drug resistant tuberculosis test that is made from steps of extraction, amplification, and detection. And here you can see how it works in details. DNA is isolated from the specimen, that is sputum, then it's amplified, then it's bound on a paper strip using sequence-specific probe immobilized on that paper strip. And then you have, of course, several lines for positive control, and so that you know the test has worked, and for the specific sequence detecting tuberculosis. So it was good. The test, they say, it gives results in approximately 2.5 hours, which is a little bit underestimated, because if you read the actual paper, they say that alone the binding to the paper stripe takes 5 to 7 hours. So I don't know from where this number comes, but it's from their official website. So the price per test, $35 to $80, is still a lot for just a paper with immobilized sequence of DNA that is supposed to bind the specimen DNA and detect tuberculosis, right? So we are still far from ideal. So what makes PCR tests so expensive is the need for special equipment of these thermocycling machines, and that's why isothermal amplification could be a solution to overcome this challenge. Here I listed 3 examples of isothermal amplification that were most cited in the recent 5 years. Recombinase polymerase amplification, loop-mediated amplification, and rolling circle amplification. Now let's talk about those a little bit in details. So there are tutorials of how this recombinase polymerase amplification works, and this is the link where you can look it up. I will try to see whether I can go to this. So how to overcome the thermocycling challenge? You have double-stranded DNA, and you need to turn double-stranded DNA into single-stranded, so that primers hybridize, and you get from 1 copy 2 copies, from 2 copies 4 copies. This green dots is called recombinase. So basically, instead of heating up double-stranded genetic molecule, they implement enzyme. Enough of this. I'm going back to the presentation. So they implement the enzyme called recombinase, this green one, that pulls double-stranded DNA into local single-stranded part, so that the primer can bind. And for one double-stranded molecule, you can get like extension of the primers. After extension of the primers, you can get double-stranded molecules, and then the same repeats. Recombinase will bind to the newly generated copies, so the primers can bind again. So you get 4 molecules. It's just like PCR, but this kit that they developed is also quite pricey, but they optimized all the concentration, all the buffer source conditions, so that you just mix your genomic DNA with a kit and run the test. But again, is a good alternative to PCR, but in science, we would like to move away from kits, because kits is a black box. And when the company will go bankrupt, what will you do? So another one here is loop-mediated amplification. It's a slightly different approach, and it will form, the product will be these complex shape structures. And I will also just press this YouTube link so that it's best to show you the video. So that's you can get from New England Biolabs. So target DNA can be navigated as certain regions named F1, F2, F3, B1, B2, B3, and their complemented regions. And these sequences that are originally present in the genomic DNA are used to create primers, forward primers and backward primers that will bind the genomic DNA, but in a fancy way. Yeah, the key phrase here is strand displacement. Whereas stack polymerase that is used in PCR is just a regular polymerase, here BST polymerase is used, and that one is known to be able to displace, like to unzip double-stranded DNA while synthesizing the copy. And this is what you see here. Another key phrase here is that these primers are designed to create self-hybridizing loops. That's why it's called loop-mediated amplification. This dumbbell structure is, let's say, milestone of LAMP. Now, once this is formed, the process will be exponential. Yeah, and then strand displacement and self-looping is the key to success. The main disadvantage, in my opinion, is that it's super complex. First, it's complex to understand, and second, it's complex to design the primers. Basically, there is no software, as far as I know, that designs these primers, unless, no, you can develop yourself if you program R or Python, but there's nothing like a user-friendly software where you can just put the genomic sequence and get the primers. For PCR, they do that, but for this one, it's a lot of pain. And another disadvantage, I mean, it's exponential, fast, and efficient technique, but it operates at 65 degrees Celsius. So it's isothermal, but 65 degrees is a high temperature, I would say. And then at this temperature, the risk of leaking and evaporation of the system where amplification takes place is higher than, let's say, at 37 degrees. Once the solution of containing LAMP amplicons has leaked, you are in trouble because the lab gets contaminated and you have to change the lab or change the primers. So once it's contaminated, is lots of pain to like redesign and move around the labs. I mean, I went through this, and many people, if you look on the ResearchGate, many people write that the technique is good, but it's very, how to say, I doubt that low-skilled technician, you know, placed in a tent will be able to not contaminate it and to do all of that, you know. The third one is rolling circle amplification, containing, made of 3 steps. The red one is a target. In this case, it's single-stranded, but it can be also double-stranded. Then you just need to make a heat shock to destabilize double-stranded DNA and then cool it down quickly so that the single-stranded parts are exposed. The black part is so-called padlock probe. The padlock probe has a light gray part that sequence specifically will bind to the light red part of the target molecule. And the sequence you know, so you can design the padlock probe binding specifically to your target. Once it's bound, a ligase is used to link the 3 and 5 prime to the probe so that the full circle is joined. And the enzymatic amplification maintained by 529 polymerase, also with strand displacing activity, just going around the circle so that you get a massive single-stranded outcome, which can be detected using plenty of readout possibilities. This is, please note, this is linear amplification. So whereas PCR, recombinase polymerase amplification, and loop-mediated amplification are exponential, rolling circle amplification, as it is presented here, is linear. This has advantages and disadvantages. Advantage is you will never get contaminated your lab because linear amplification does not create so many, it cannot amplify from a single copy. But the disadvantage will be the sensitivity. We will speak about it in later slides. But for now, I presented you like the big picture of diagnostics, different targets, and why nucleic acid test is needed. And talking about different readout strategies, I would like to introduce you optomagnetic biosensor. So it's developed by these 2 people at DTU, Fock and Hansen, and it's made of magnetic nanoparticles in oscillating magnetic field, which create a so-called shadow of light passed through the suspension of these magnetic nanoparticles. So the particles, they try to align in the magnetic field, and if they are aligned with the light, then the signal will be detected. If they are not aligned, then the signal is 0. So this is what you basically see here. This is a magnetic field. This is a sinusoidal magnetic field. And when the field has a maximum intensity, then the particles are aligned and you get the signal, optomagnetic signal. Yeah. This optomagnetic signal is second harmonic because it doesn't matter whether the field is positive or negative. And now I'm going to explain you a little bit more in details. So you see the sinusoidal magnetic field and the optomagnetic signal, second harmonic optomagnetic signal. And you see these 2 magnetic nanoparticles. Now, if we apply a magnetic field of low frequency, then the particles have time to fully align with the field so that they align and the light will just pass through the suspension and will be detected large optomagnetic signal. But if I will use a higher frequency, then the particles never really align perfectly. And if I use a very high frequency, they almost don't move. And of course, they are chaotically distributed and they block light. They only pass the light and cause optomagnetic phenomena if they are aligned with the magnetic field. That's why at low frequency, you can see the optomagnetic, the light pass through, and at high frequency, you don't. This is what basically this spectra show. And then, yeah, I will not go into details through the math, but the details can be found in this paper of Jeppe, characterization of fine particles using optomagnetic measurements. What I want to say only is that optomagnetic signal can be presented as a function of real part and imaginary part, where real part is the sinusoidal component and the imaginary is a cosine function. And there is also always a phase. It's a delay between magnetic field and the rotation of the particle. And this phase can be also used for analysis. So here you can see the plots of real part, imaginary part, and the phase. When phase is equal to pi divided by 4, then the imaginary part is 0. And this frequency, when the imaginary part is 0, is actually equal to the Brownian relaxation frequency that you can find inversely proportional to the hydrodynamic volume of the nanoparticle. Again, all the math you can find in this paper. But this is very useful information that you can get from optomagnetic measurements. The hydrodynamic diameter of magnetic nanoparticle as function of, for example, DNA targets that you would like to detect. Now, after I gave you introduction of so many things, we move from theory to practice and directly how measuring hydrodynamic size as function of target DNA concentration can be beneficial for diagnostic purpose. So yeah, indeed, this is loop-mediated amplification that I called very complex, but it was the first thing I did at DTU. So we can create a forward primer with biotin group and bind it to magnetic nanoparticle via biotin streptavidin complex, which is the strongest in the nature. So now primer is attached to the particle. And remember, in course of LAMP, the size of DNA is getting bigger. Size of DNA attached to magnetic nanoparticle is getting bigger. So the frequency is irreversibly proportional. So frequency going to decrease. This is what we see. Because of amplification, the peak of optomagnetic signal is shifting to lower frequency. Now we can use MATLAB to find the maximum of the peak and extract the hydrodynamic diameter using this formula. And indeed, it is a function of the target concentration. The target that binding the FIP, the primer, as well as other 4 primers, getting elongated, and the hydrodynamic diameter is increasing from 130 to 280 nanometers. But this happens with a different threshold time. Remember from PCR, it's not PCR, but it's a similar principle of exponential amplification. The more target is present in the solution, the earlier the amplification will start. So for 100 picomolar, it starts after, say, 5 minutes. For 1 femtomolar, it starts after 20 minutes. The negative control was negative. You see, there was no change. But in some cases, we saw deviation here after 40 minutes. So I said it's dangerous. So we decided to set threshold time 20 minutes. And we can say that, okay, somewhat between 1 to 10 femtomolar of dengue RNA can be detected using this technique. But soon our lab was contaminated. So this solution wouldn't be long lasting. I mean, it's definitely enough to publish a paper, but it's not what we want, right? We want to create a solution for good. What else can be done using mediated amplification? So we decided to discriminate or differentiate so-called false positive and the true positive amplicons. For this purpose, the negative control and the samples containing different target concentrations were run to produce the amplicons, and then they were captured on using these loops. The magnetic particles containing sequence complementary to the loop of originating from dengue DNA. And if a spurious amplification takes place because of the nature of the polymerase, BST polymerase is not highly precise. So it likes to introduce wrong bases, DNA bases. I told you that DNA is made from 4 bases, and during amplification, mistakes happen. So, and if the amplification is recycled many times, what usually happens with the spurious amplicon, then the sequence matches, but with many gaps that called mismatch. So after binding at 40 degrees, we can see that the signal is almost same. But if we hit a top at 67 degrees, we see that the true positive DNA amplicon is still attached to magnetic nanoparticles, whereas the spurious one containing errors and mistakes and mismatches is getting detached. In fact, you can do the melting assay, and you can see that the melting point of the spurious amplicon and the positive amplicon is highly different. It's from like 58 or 57 degrees for the spurious, for the false positive, and somewhat around 75 for the true positive amplicons. But we were rather unsatisfied. I mean, we published it in this analyst paper, but we later moved on with the rolling circle amplification. That is isothermal, and that is a good alternative to PCR, but unlike LAMP, it's more controllable and probably will be a real solution to the problem. Okay, so now I will be talking about development, integration, and multiplexing of linear rolling circle amplification, as well as genotyping of CAT-G gene of Mycobacterium tuberculosis. So I told you that CAT-G gene is the one responsible for antibiotic resistance if mutation happens. And this result, whatever I will present from this topic, is part of this publication here. So I must acknowledge a contribution of BlueSense Diagnostics at a startup from DTU that developed Blu-ray and lab-on-chip solutions, of course, together with DTU. and this optomagnetic technique. And here we speak about assay integration. So we move from just pure amplification techniques to how the steps can be integrated and automated. And since it's isothermal, it can be implemented in a low-cost plastic chip, such as this here is made from PMMA. And then you can take a Blu-ray system with this laser and put the electromagnets and use the optomagnetic principles to detect DNA in this system. It's highly robust. The discs are made from 3 layers of PMMA using pressure-sensitive adhesive. I did many of those myself. And depending on the application, for example, if you study clusters of magnetic nanoparticles like here, permanent magnet can be placed to accelerate particles, because electromagnet is just causing sinusoidal magnetic field. Permanent magnet is really applies a force to bring particles together, like literally increase concentration of them so that the binding takes place faster. So what can be done with the disk? Together with my master student, we improved the disk design so that it would contain 2 chambers. One chamber is for ligation. Another chamber is containing the particles, nucleotide phosphates, and polymerase called Equify. For 20 minutes, we perform ligation. Then we spin down. Of course, everything is automated using LabVIEW, so that 2 mixtures come together. You can also mix it by quick rotations left and right. And then we allow amplification to take place. So what happens? You remember I told you that the optomagnetic signal is made from real and virtual imaginary parts. In a real part, you can see initially the blue, wait a second. The very first blue spectrum is a spectrum of just magnetic nanoparticles when they are single. In course of amplification, the changes happen in a way that this population is depleted from blue to red. And the population frequency about 20 hertz is built up. At 20 hertz, hydrodynamic diameter is significantly reduced, and this corresponds to the clusters of the magnetic nanoparticles bound to the product of rolling circle amplification. Appropriate changes can be also seen in the imaginary part. But for the analysis, we chose to use the phase, that is basically arctangents of these 2. And the phase is 0, this frequency range is the beginning, then it's significantly decreased. The phase is decreasing means the particles lag behind the field. And as big as the clusters get, as more this lag phase can be detected. And then we read the signal in this frequency range by integrating the curves for the strain that has a mutation. So the red one is antibiotically resistant. The wild type that doesn't have mutation, so this one is not antibiotically, it's not resistant. And then no template control. You can see that it's highly specific. And it, in this case, it's of course qualitative. It's like yes or no. But let's see whether we can do any better. I also would like to present another strategy that we compare between. In this case, we place, it's not a disk, it's just a single one-chamber chip made using the same approach from PMMA layers and pressure-sensitive adhesive. And these chips are placed in this optomagnetic setup made of the electromagnets, photodetector, LED with the same wavelengths. So it's just same. The only difference is that here the temperature is made from aluminum plates, which is contact. It comes into the contact with the chip, like the hot air flow in this box for the disk setup. So the temperature control is much better. But apart of that, you see, it's also, ligation takes place, then the temperature is shifted so that polymerase is activated. We see the amplification and clustering. Same analysis, same outcome. We can sequence specifically detect the strain with mutation, whereas the strain without mutation gives the signal equal to negative template control. So let's see what can be done with these two approaches. We spiked the different target concentrations in 5% fetal bovine serum to mimic the conditions, because serum contains protein and some ions which can potentially reduce activity of the enzyme. It might also have proteases, I'm not sure. But this is a good way to see how the test will be working in a real condition. And these are called dose response plots. So you see that the lag phase is increasing from 0 to 60 picomolar concentration. And the limit of detection, it can be described differently by different people. We decided to describe it as the lowest concentration that is of the cutoff value. And the cutoff value is just a negative, no template control plus 3 standard deviations. So you see that in this case, the D is 2 picomolar. Although one can argue that it's rather, I don't know, 5 or so, but using our description, it's 2 picomolar. And this is for one chamber chip. And for the disk setup, it's 5 picomolar. Now we can compare them. The limit of detection, whether it's 2 picomolar or 5 picomolar, is really very similar number. But what's important is that the total time is, in this case, 65 minutes, in this case, 95 minutes. So of course, it's shorter using one chamber chip. But if we speak about multiplexing, the disk has 16 pools. The single one chamber chip setup has only 4 positions where we can place chips. So the throughput of the disk is higher. The main disadvantage of the lab-on-a disk setup was that the temperature airflow requires a longer time to stabilize, so it's also overshooting, and then it's cooling down. So it's the precision is very bad. It was very bad, but it can be improved. I mean, it was just a master project. What's important to remember is what we are doing is the integration of the assay is when the person, the low-skilled technician, can mix solutions together, place them in a system, press the button, start, and get the answer. So in this case, he wouldn't need to digest what went wrong, and and this is compared, for example, to non-integrated assays. So we got this 2 or 5 picomolar LOD, and using fluorescence readout, Gao et al. Claimed that they can detect down to 10 picomolar target, and Pavan Kumar et al. Claimed that they can detect something around 30 nanograms of tuberculosis DNA. And 30 nanograms, I calculated, is about 0.5 picomolar in 20 microliter solutions that they have used. So it's a similar sensitivity. Indeed, this paper microfluidics is pushing the market. So Pavan Kumar established, together with other people, this MPE diagnostic company, and they, I assume, put on the market this product that would be able to detect these sequences. But remember that it's not integrated, which means they do the ligation step, amplification step, and then they have to put it on this chip. So all of these steps, they need to be taken outside in the laboratory, and then they are tested. So where are we? Where we are? Our assay is supposed to help to discriminate, to find the tuberculosis and to discriminate strains that have antibiotic resistance from the strains that do not have. And instead of 6 to 8 weeks, it's supposed to take 65 or 95 minutes. However, our best limit of detection, well, 2 or 3 picomolar, is that corresponds to 2, I would say it's a trillion cells in milliliter. So this number is extremely high, but this number is equal to 1 colony. So what can be done right now is that they can take a colony, swab from the colony, and use our assay to figure out whether it's resistant to antibiotic or not. So it can be used for genotyping, but it cannot be used just to detect tuberculosis from sputum, because I doubt that sputum will contain 2 trillion tuberculosis cells. Why is it like that? Because we are limited by linear amplification rate. How can we boost the amplification rate? Wait. So here you can see it's a very similar rolling circle amplification strategy, but it has these scissors here. And scissors represent the nuclease that cut that nick, create a nick on a newly synthesized strand only because we introduce methyl group here. Okay, so it's not a full cut, it's only one-sided cut. And if you create cut, then more polymerases can be attracted because 529 can start strand displacement also from the nick. So while this polymerase is already on the way to synthesize a strand, another polymerase sits here and starts. This results in many polymerases working on one circle, and this can, of course, boost. It's still linear, but it's by factor, I don't know, 5 or 6 better than just what I have presented before. So my colleague Bo at DTU, he has employed the same approach using phase lag strategy. And it seems like he could claim 32 femtomolar detection of the tuberculosis DNA, which is by factor 100 better than what we observed. But remember that he didn't do integration here. So he did the steps without putting them together. The outcome of his work was that nick RCA, what he called it, is by factor, I don't know, yeah, by factor 100 is better than conventional RCA. He used 90 minutes, and he could detect down to 30 femtomolar. Then when he added the 10% fetabulin serum to mimic the real conditions, of course, it messed up a little bit, so the limit of detection is increased, and yeah, you can see it's somewhat below 100 femtomolar, but depends how you see the limit of detection. I think he projected it as his point that crosses. He didn't do precise titration in this range. But anyway, there was a significant improvement using NIC-RCA. And I remind you that we still pursue detection of multi-drug resistant tuberculosis bacteria using RCA and optomagnetic readout. So what else can be done? Else we can implement a post-RCA amplification. So this is our latest publication. Again, this is a target ligation and an amplification with the nuclease, so that many polymerases can act one circle. Then there is you get intermediate amplicon that can bind an extra template here using the same nuclease off-loop twin displacement amplification creates end amplicon, and that end amplicon is used to link two magnetic nanoparticles together so that we can detect clusters. Let's see how that worked. 10% fetabulin serum after 90 minutes of reaction. Or I guess I use the same screenshot. Yeah, but this is a publication, I believe. My bad, but here you can see I have a publication opened. So he used 90 minutes of RCA followed by 50 minutes of this stand displacement amplification. And in 10% of fetabulin serum, the limit of detection is 30 femtomolar. Let me, I want to, yeah, so we've got from around 100 or so to 30. But personally, I think that the further improvements, they fade because you have to introduce more and more steps for low gain. But let me wrap it up. Today I presented you as that optomagnetic biosensors developed at DTU and say together with the BlueSense Diagnostics. It can be portable, and indeed that company, BlueSense Diagnostics, has launched the ViroTrack, a product for COVID-19 point-of-care diagnostics using magnetic nanoparticles. So it is portable. It has a potential for point-of-care diagnostics. Isothermal RCA assay is integrated in a plastic chip, and different variants of RCA to overcome linear amplification rates are compared. The ultimate detection limit of 100 femtomolar or even below down to 30 femtomolar is achieved in 10% fetal bovine serum. It's still high number of cells, but we are on the way to get closer. And even if not, I mean, if we can somehow shorten the number of the weeks the technician has to wait until the colony grows, yeah. So even if it's 2 weeks less, it will also help. For the last perhaps 10 minutes, if I have, I will just give you an entertaining slide of what else can be done using nucleic acid probes for diagnostic purposes. I mean, what people research on. One very nice work I found was detecting mercury using nucleic acids probes. So in this case, nucleic acid single-stranded DNA is folding in the presence of mercury, and the fluorescent dye cybergreen intercalates into folded DNA and lights as a green signal, whereas when it's binding single-stranded DNA, it's yellowish. So of course, it's just a picture, but what they did the dose response analysis using fluorescence spectroscopy, where you can have, emission filters to cut the yellow fluorescence and get a nice signal analysis. The electrochemical biosensors, they are also quite promising, although they are less attractive for point-of-care solutions. The cleanliness standards for electrochemical reactions, they are sometimes too high. But in this case, there was HIV RNA, that's a single standard, was detected using Tuberculos probes. Yes, DNA can be folded in a triplex form, and what's called molecular beacon, the single stranded DNA with the electroactive probe, when it's unfolded, has a low current detected. But when in the presence of the target, a hairpin structure is formed, bringing this electroactive molecule close to the electrode, and the current is much higher because of the vicinity here. This is how they detected it. Gold nanoparticles is a classic example for the naked eye detection, where you can link the gold nanoparticles and see the transition from red to blue, I believe, from single to the clustered gold nanoparticles. This paper, diagnostics, as I was talking, is I just put it back here to say this is my picture of what people are working on. They use different readouts, fluorescence, electrochemistry, gold nanoparticles, paper-based, and we have our optomagnetic biosensors that I believe found a place in a row of these other well-known techniques. I would say I am done with my presentation, and thank you so much for your attention.
Thank you. So, thank you, Antonio, for your nice presentation. Yeah, I guess.
Yeah.
Muted. Thank you, Anthony, for your nice presentation. Actually, you did a great job, and you invented a very nice system for detecting of different kind of diseases based on nucleic acid.